Welcome to clickmyproject.com. Our project title is Learning Phenotype Structure Using Sequence Model. The abstract of our project is a microarray is a collection of small DNA spots attached to a solid surface. In microarray experiments, the signal collected from each spot is used to estimate the expression level of a gene. A microarray contains thousands of DNA spots. An essential problem in microarray data analysis is discover phenotype structures. The objectives are discovering groups of samples. For each assembly of samples, discover the typical expression patterns. The collective drawback is that the recognized signatures often include a large number of genes but with low discriminative power. Here we propose a model is G-star sequence model to report this constraint. Let's go for data flow diagram. First, our process is the data mining. So here we have to load the data set. After loading the data set into the database, we have to pre-process the data. After that, we have to compute the similarities, that is the occurrences of the data, and finally we have to evaluate the performance using matrix computation. After that, we have to classify the malignant and benignant. Now I am going to show the demo of our process. First, run from home.java. Here we have to choose the data set for data mining process. This is our data set attributes. Next we have to load into the database. These are the attributes of the particular data set. Next we have to pre-process the loaded data. The data pre-process which is successfully finished. After that we go for sampling. We are just sampling the values as 1 and 2. Next we go for standard deviation. First we have to here choose the attributes of our data. These are the selected attributes in our process. Next we have to find out the standard deviation. Next we have to construct the entropy, conditional entropy and relevance. First we are going to find out the entropy. This is the entropy values. Next is conditional entropy. Here relevance which is computed. Next step is similarity computation. The next step is matrix computation for each features. Here we have to find out the matrix for each representatives. So it matches with one another. From that we can define the matrix. This is the final similarity between the attributes. Next we are going to cluster the attributes. We are just clustered it as a 1 and 2. Now the cluster 1 values which are selected with their ID. This is cluster 1 and this is the ID for cluster 1 and for cluster 2. Next is the final step, which is the signature detection, which is we are going to classify the malignant and benignant. This is the most affected data IDs, which is classified in the cluster. Next is benignant values. Finally, we have to evaluate the performance of existing and proposed work. This shows the accuracy of the proposed system. Next is the discriminative power signature. Here it shows the existing system and it shows the proposed system of the signature. Thanks for watching.